Our next speaker is Franco Felix from Stanford, who's going to talk about grainy head-like 2 and AP2A in the surface ectoderm.
Hi, good afternoon, everybody. My name is Franco Felix, and I am a third-year PhD student in the Stem Cell Biology and Regenerative Medicine PhD program over at the Stanford University School of Medicine. And I'm here to tell you a little bit about our work in the lab of Dr. Anthony Oro in the Department of Dermatology, specifically how these two transcription factors, grainy head 2 and AP2A, can coordinate surface ectoderm lineage commitment in our induced skin composite differentiations, which create epithelial keratinocytes, the major cell component on the outer layer of the skin, or the epidermis. And so with that, the Oro lab is interested in producing a cell and gene therapy for the treatment of epidermolysis bullosa, which is a disease that under minimal trauma to the skin causes blistering, erosion, and scarring of the skin, which leaves the door open for the development of lethal squamous cell carcinoma. And so we've really spent the last decade working on creating a clinical manufacturing protocol where we can take patient fibroblast samples, reprogram them into pluripotent stem cells, and then genetically correct the collagen 7A1 mutation which causes this disease, and then differentiate those pluripotent stem cells into induced skin composites, which contain these epithelial keratinocytes. And we've heard a lot of amazing talks today that talk about the complexities of creating a clinical manufacturing protocol and everything that goes into them being approved by the FDA. And for this talk specifically, we can see this very complex schematic of the clinical manufacturing protocol, but myself and the postdocs that I'm going to talk about today are interested in the actual transcriptional and epigenetic networks which drive lineage commitment in these induced skin composites. So to introduce the induced skin composites, we start out with pluripotent stem cells, and we differentiate them for 45 days, composed of an initiation and a maturation phase. By the 45th day, we get several different cell populations. We get mesoderm-derived fibroblasts, surface ectoderm-derived epithelial keratinocytes, and neuroectoderm melanocytes, or neural-like progenitor cells. At day 45, we can enrich these by sorting for the epithelial keratinocytes and then engraft those onto mice, in which here in this immunofluorescence imaging, we see very nice stratification of these epithelial keratinocytes. Stratification just being creating these several layers that we see on the outer layer of the skin, or the epidermis. And the reason we've spent so much time, about 10 years, working on this differentiation protocol is because we really want to increase the robustness. So being able to have several different personnel in different facilities consistently get the same outcome in this differentiation. We also want to increase the efficiency. So can we produce 70, 80, 90% of the cells becoming keratinocytes, which will really save us in the costs of manufacturing epithelial keratinocytes? And also, can we increase the functionality of these epithelial keratinocytes, essentially their ability to engraft and stratify, not only in these mouse models, but hopefully in the future patients. And I think we can, I think this is the wrong talk. This should not be what I have on the Google Drive from last night. Yeah, 1 second. If not, I can wing this in a second. So. Sorry, everybody. I switched my slides probably a little too last second, just to make it I think, a more straightforward presentation, but we'll see. Okay, great. Here we go. So the reason I would like to focus on these, we've gone through this induced skin composite differentiation protocol. We've seen that these keratinocytes have the capacity for engraftment and stratification on a mouse. We want to increase the efficiency, the robustness, and the functionality of these keratinocytes. And I really believe we can do this by focusing on day 7 of our induced skin composite, so after this initiation stage. The reason being is that these pluripotent stem cells are just now beginning to commit to the different lineages that are exhibited here in this induced skin composite. Specifically, what we'll be looking at is how GRHL2 and AP2A coordinate surface ectoderm lineage commitment and inhibit that of the neuroectoderm. So GRHL2 and AP2A are 2 transcription factors which have been shown to be critical for epidermal development in murine knockout models, but the mechanism by which they do this has been understudied to this point when we started working on this project. And so postdoc in the lab, Annie Collier, created a GRHL2 knockout cell line of pluripotent stem cells. You can see the depletion of the GRHL2 protein in this Western image below. And when we look at day 7 induced skin composite immunofluorescence, we see in purple that the wild type GRHL2 levels are depleted in the knockout, and we exhibit keratin 18 disorganization and protein depletion in the GRHL2 knockout, keratin 18 being an essential marker of development in our epithelial keratinocytes. If we overlay that with the DAPI imaging, you see even a more exacerbated keratin 18 phenotype. And so with this, we're really happy to see this result, which indicates to us that GRHL2 promotes surface ectoderm lineage commitment in our induced skin composites. And I'm going to skip over a lot of the preliminary work that was done to get to the next stage, just so that I can show some pretty cool data. So I conducted a single-cell multi-omics on these day 7 induced skin composites from wild-type and GRHL2 knockout cells. And what single-cell multi-omics is essentially you're taking each individual cell and you're producing both ATAC and RNA-seq libraries. ATAC-seq essentially is looking at chromatin accessibility. So whether a gene and its regulatory elements are accessible, and the RNA expression library is looking at RNA transcript abundance, so whether that gene is being expressed or not. We then can combine these 2 modalities, ATAC-seq and an RNA-seq, to plot the cells on a UMAP, essentially indicating how similar the cells are and creating populations. If we look at the UMAP of day 7 induced skin composites with integrated wild-type and GRHL2 knockout cells, we see that we get the 3 populations that I spoke about earlier, mesoderm, surface ectoderm, and neuroectoderm, and even this more advanced neural population that is composed of purely GRHL2 knockout cells. If we then color code this UMAP for wild-type versus knockout cells, and we quantify the number of cells in each population, we see that the GRHL2 knockout cells are producing far fewer surface ectoderm cells and many more neuroectoderm cells. And so with this, I won't talk too much about these analyses that we did with the single-cell RNA, but we looked at gene expression, pseudo-time analysis, essentially how mature, immature these cells are in comparison to one another. And we've also done a lot of work on the single-cell ATAC, looking at transcription factor motif accessibility, indicating that one or another transcription factor may be active in this differentiation, and also linkage plots, so whether chromatin accessibility correlates with the expression of a certain gene. But for the main point to take away from this multi-omic experiment is that GRHL2 is promoting surface ectoderm gene accessibility and transcription while inhibiting that of the neuroectoderm. So we really want to know how GRHL2 is doing this, and so we decided to do chromatin immunoprecipitation sequencing, or ChIP-seq. Essentially, we're breaking apart the DNA and then pulling down GRHL2 and any DNA that it's bound to. We can send this for sequencing, and we'll get hits throughout the genome of where GRHL2 is bound. If we then look at GRHL2 binding in respect to different histone modifications to indicate regulatory elements, we see that GRHL2 binds strong enhancers and active promoters. And if we do module scoring, this violin plot below, we see that GRHL2 binding is greatly enriched at surface ectoderm genes and not so much that of the mesoderm or neuroectoderm populations. And so with this, we've identified that GRHL2 binding is what's facilitating the accessibility and the transcription at these surface ectoderm genes while suppressing the neuroectoderm lineage commitment. But transcription factor stories are rarely ever so simple. They typically work in complexes, and this is where another postdoc in the lab, Gillian Pattinson's project, very much complemented this work on Granny Head 2. So she created an AP2A knockout cell line, and this first Venn diagram above looks at genes that are dependent on Granny Head 2 or AP2A in our induced skin composites. And we see a very large overlap of dependent genes on these two transcription factors. If we take this a step further and look at Granny Head 2 and AP2A bound sites, we see that they also overlap with a large significance. And so this indicates to us that these two transcription factors may be cooperating together to regulate these genes. And so we were interested in, is Granny Head 2 important for AP2A localization at these surface ectoderm genes? And so we did an experiment where we did AP2A ChIP-seq in wild type and Granny Head 2 knockout cells. And here in the tornado plots below, we see very nice wild-type binding for AP2A. But in the Granny Head 2 knockout, AP2A binding is all but lost, indicating that somehow Granny Head 2 is important for the localization of AP2A to these different genes. And below the tornado plot is just a track that shows wild-type versus Granny Head 2 knockout AP2A binding at the EpCAM gene, a very important surface ectoderm gene. If we then do the reciprocal analysis, so looking at Granny Head 2 ChIP-seq in AP2A knockout cells, here the wild type, this is a baseline, so zero binding. We see that Granny Head 2 is gaining 5 to 6-fold binding in the AP2A knockout. And so this is indicating to us that AP2A has almost a completely opposite role on Granny Head 2. It's actually inhibiting ectopic Granny Head 2 binding, which we know scores for neuroectoderm genes. And so with this, we've determined that Granny Head 2 binds and facilitates accessibility and transcription of surface ectoderm genes, while also promoting AP2A binding to those surface ectoderm genes. AP2A then turns around and inhibits Granny Head 2 ectopic binding to that neuroectoderm gene network. And this is how our surface ectoderm develops and commits in our induced skin composites. And now the question is, how do we use this transcriptional network to enhance our differentiation, increasing the robustness, the efficiency, and the functionality of these keratinocytes? And the answer may be in the Granny Head 2 and AP2A binding network, in which they regulate the ABCA4 and RGAP29 locus. And RGAP29 is an activator of the Rho pathway. And the Rho pathway can, in part, this is a very partial Rho pathway here, but the Rho pathway can be in part activated by mechanical cues. So the stiffness of our tissue culture plates, the topography of those plates in the extracellular matrix, or even cell density. And many of us here will recognize the Rho pathway because we treat our stem cells, our pluripotent stem cells, with ROCK inhibitor after single cell dissociation and seeding. And this pathway really regulates the actin cytoskeleton, intermediate filaments, and focal adhesions, which are incredibly important for these surface ectoderm keratinocytes. And so the question now is, in this differentiation protocol, we're treating the cells uniformly with the same initiation and maturation media, and yet we're getting several different populations that develop. And how is this happening? One answer may be the spatial organization at day 7, which these induced skin composites exhibit. And this was supposed to be in some of my earlier background slides, but I'll introduce it now. In red here is RNA-scope imaging for surface ectoderm markers, in which we see surface ectoderm develops in these nice circular colonies, which are flattened out and very sparse. If we look at this green RNA-scope image, these are mesodermal fibroblasts, and we see that they tend to encircle these surface ectoderm cells, and they're very compact, a very high cell density. And we believe this is important for the crosstalk that is exhibited in development between the epidermis and the dermis layer, and that this might be facilitating higher levels of engraftment and stratification capacity for epithelial keratinocytes. And we believe that this might be influenced by this Rho pathway, so it's possible that these mechanical cues are then taking over these transcriptional networks from AP2A and Granny Head 2 that we've spent so much time talking about. So a lot of the future directions in the lab is focusing on how mechanical cues can be used to regulate these different transcriptional and epigenetic networks for the different cell lineage commitments in our induced skin composites. So with that, I would just like to give a summary in which we've Talked a little bit today about how GRHL2 and AP2A coordinate surface ectoderm lineage commitment by binding to surface ectoderm genes, while AP2A inhibits GRHL2 binding to ectopic neuroectoderm genes. And our future directions will focus on how mechanical cues that play a role in this transcriptional network may be used to produce induced skin composites with a higher robustness, efficiency, and higher functionality of these keratinocytes to stratify, engraft and stratify in the mouse models or hopefully in the future patients. So with that, I would like to give a huge thank you to my lab, especially my PI, Dr. Anthony Oro, and Annie Collier, the postdoc who I worked with on this project. I forgot to add a slide, but this work, some of this work has been published with Annie Collier as the first author, so please give that a read. And I would also like to thank the CIRM organizers for organizing this phenomenal conference, especially thank you to the tech team. You guys were able to solve that problem very quickly. And then also thank you for the funding that CIRM provides. So myself and several of the other Stanford trainees that are here have fellowships from CIRM, and so without this, our work's not possible. So thank you for everybody for attending this talk. If the speed of the talk or any of the jargon I use confused you, I apologize, but please feel free to ask any questions or approach me later on at the reception. So with that, I'm happy to take any questions. All right, no questions. Thank you, everybody.